Hello, everyone, and welcome back to Microbiology. This lecture will cover Chapter 3, the cell. This lecture will assume that you already are equipped with an understanding of the four biomolecules of life, that being sugars, lipids, proteins, and nucleic acids, and that you know what the basics of mitosis are. The lecture objectives are to compare prokaryotes to eukaryotes, learn about different morphology and cell arrangements, understand the effects of osmosis on cells, and learn about different parts of the cell and their function. There are two main types of cells, prokaryotes and eukaryotes. In previous biology courses, you probably learned that prokaryotic cells are smaller and simpler than eukaryotic cells, and that the most important difference between them is that prokaryotic cells lack a nucleus. A nucleus is an organelle, which, if we think of our bodies having organs, it's kind of similar on the level of cells. Cells have smaller functional units within them called organelles. Depicted in this image, we can see the DNA is surrounded with a lipid membrane, which is the purple dashed line, and it houses the chromosomes, or genetic information of the cell. Within biology, there are two major classifications of cells: the prokaryotes, from their words meaning pre or pro nucleus, and eukaryotic, where eu means true, so true nucleus. Cells of microbes can be either of the two. Viruses are neither prokaryotic or eukaryotic, and aren't even technically made of cells. We'll discuss them later in the course. This classification is based on how the cell stores its DNA, but because DNA is the blueprint of life where information is stored, how that information is stored, we will see has profound effects on how it is used. The prokaryotic lineage is the oldest, with pro meaning pre or before nucleus. Essentially, their DNA is stored in a generalized location without any membrane surrounding it, and this location is called the nucleoid, not a nucleus. By the way, if your prerequisite course is a little fuzzy, take a moment to review what a cell membrane is, what it is made of, and what it does. The eukaryotic lineage has a membrane that encompasses the DNA. This structure, seen as the purple dashes, again is called the nucleus, and an example of a eukaryote are us humans. An example of a prokaryote are E. coli. Careful to not let this confuse you. As discussed previously, eukaryotes can be single-celled microbes as well, for example, yeast. Here we list a few major differences in the way prokaryotes and eukaryotes conduct things. Prokaryotes and eukaryotes can be divided amongst other traits as well, of course, but the DNA location being the most obvious. Prokaryotes contain a nucleoid, which means nucleus-like, and it is an irregularly shaped region within the cell of a prokaryote that contains all or most of the genetic material. The core genetic material of a cell is called their genome. Chromosomes are another name for the structural organization of an organism's DNA. In prokaryotes, it is circular, meaning it has no end. However, eukaryotes are not circular. They come in linear segments that have a beginning and an end, and they are not bound to each other. An organelle is a compartment inside the cell that serves a function that is surrounded by a membrane. The first example of this we have discussed so far is the nucleus, but eukaryotes have many of these organelles, while prokaryotes don't really have them. There are some unique exceptions, but in general, eukaryotes have many organelles, prokaryotes do not. Prokaryotes have a strong material in their cell walls that is called peptidoglycan. That lends to the strength, structure, and shape of the cell. Depending on the eukaryote, they may not even have any extra cell wall material, just a membrane. But some of these eukaryotic organisms have chitin or cellulose or other material as well. DNA packaging is extremely important. Depending on the organism, DNA can range from millions to even billions of individual units bound together that make up the genetic code. The cell needs a way to package that information neatly. Eukaryotes have some of the largest genomes, and they have proteins scientists call histones that wrap up or package the DNA. Look at this image from right to left. We can see the unpackaged double helix DNA first, and then as we moved to the left, we can see how it's organized. Histones are used to wind up the DNA into coils, and then those coils are coiled upon themselves to form what are called supercoils, which are the structure that is condensed to form the linear segments of chromosomes of eukaryotic DNA. So if you look all the way to the left, you can see a typical karyotype or chromosomal structure found in eukaryotes, and notice there are ends to these chromosomes. And with a prokaryotic cell, it's just one big circle. Finally, prokaryotic replication occurs through binary fission, whereas in eukaryotes it occurs through mitosis. Recall that there are three domains of life: the eukarya, archaea, and bacteria. We will focus primarily on bacteria because they are well studied. They are responsible for a significant amount and types of human disease, and because we know incredibly little about archaea and their roles with humans. We will also briefly cover eukarya, which contain the microbes, fungi, protozoa, algae, and helminths. When we later cover acellular microbes, which means microbes that don't have cells, remember A means without, we'll get into greater detail of viruses, but this chapter is about the cell. Let's begin with bacteria then. Morphology describes the form of the cell. Take a moment and try to remember why cell morphology is so important to microbiology. If you said it's important because it is necessary for the identification of microbes, then you are correct. Prokaryotic cell morphology comes in three different shapes. Coccus, from the Greek meaning round or spherical or ovoid; bacillus, which is a rod-like shape; and spiral, which is corkscrew in shape. Note in this image the coccus, which is on the left-hand side, can sometimes have the appearance of a bacillus bacterium. This is due to the fact that this is a particular bacterium going through binary fission. And if one looks carefully, they will see a little indent on the cell wall, which indicates that this cell is going through division, and those are two cells in the process of forming and separating from each other. A little note here to remember while we're talking about these words here is if the word ends in us, it is the singular form. That bacillus is rod-like in shape. If the word ends in I, it is the plural form. We can see the difference in the following example. Those two cocci may look like a single bacillus cell, but upon closer examination, they are going through binary fission. In this example, cocci is a plural form of coccus, and bacillus is a singular form of bacilli. Bacteria also have different arrangements in this image. You can see diplococci and streptococci. What determines the arrangements is the plane of division that occurs in the bacteria. The plane of division is along what axis the cell breaks apart, shown here in the blue square that intersects the cell. So that would be its plane of division. That's where the cell will divide into two more cells. If a bacterium divides along the same plane for each of its progeny, so if its daughter cells then divide along the same plane of axis here, and they do not separate, then you can have either the diplococci or streptococci. Diplococci is when they only stick together in pairs, and streptococci is when they stick together in long chains. Note the size of the bacteria we're looking at with the scale bar. These microbes are in the range of about 0.8 micrometers to 1.5 micrometers, also called microns, which is very typical for bacteria. So by looking at this scale bar, you should very easily be able to tell if you're looking at bacteria, maybe archaea as well, but you can rule out if it's an a eukarya, because it's just way too small for what you're going to find for eukarya. Streptococci can be miscategorized by students as spiral bacteria because they can make what appears to be a spiral shape when you add them all up together. But you need to look carefully and see that they are made up of individual cells. So again, we're going to be looking at the outlines here, and you can see that these are all individual cells here. They're not one smooth cell, as we may have seen in the prior slide here. See, this is just one cell. You see how the cell wall is smooth all the way across? Much different than what we see in this image here. We have individual cells. So be very clear about that, or very perceptive there. Because plane of division determines the arrangement of the bacterial cells, if a bacterium divides on two different planes that intersect each other at 90-degree angles, as in image B, this division pattern leads to a tetrad. So here we see one plane, and then 90 degrees, we have another plane. And so this cell could divide into, say, these two cells, right? And then now we're going to divide along this plane and make the two back cells. And that will create a tetrad, which if you've ever played Tetris, you should know that tetra, it means four. So a tetrad are four bacterial cells that can remain as a group. If a bacterium divides on three different planes and stick together as groups of 8, we can have a sarcane, which is what we see in image C here. Lastly, if we have a bacterium that divides without a set plane, then we can have clusters that look like a bunch of grapes. And this is called staphylo. In this example, these are cocci, so it would be staphylococci. Remember, this isn't staphylococcus because there are many cells here, so we're using the plural form. Most bacterium separate after division. In this example, here we have a single bacillus, and occasionally, and what I'm doing here is we want to make sure that you're able to tell the difference between a single bacillus and, say, a diplobacilli. The reason why is you'll often see these bacteria lingering together, but that doesn't mean it's diplococci. This just means it's going through active division. Okay? So you might see single bacillus attached to each other, but as we can see, these cells are not a full-length cell as compared to some of the other longer ones, like the one circled in red. And so this indicates that these cells are going through binary fission. However, in example B, we can see that these cells can often remain as partners, even as we see here in a full size. So these are referred to as diplobacilli. If you see in a culture that the majority of cells are diplobacilli, I mean the vast majority, and they, for the most part, look like full-grown cells. So if you see any single cells, they're not any bigger. You are likely looking at a true diplobacilli species. If you see many of them as a single bacterium, and the single bacterium tend to look larger than the bacterium together, like in example A, see how these two are much more stubby, and this is a full-grown single adult bacterium, then what you're looking at is just a bacillus species. If these bacilli remain attached by their polar regions to each other, then we can have a streptobacilli. Sometimes these bacterium are somewhere in between and have an identity crisis. So these bacterium are called coccobacillus. They are truncated. They're not a true full length of a bacilli, where they are not quite round enough to be fully a coccus, right? These down here, they're not round enough to be called circular, right? There's a lot of flat side to these ones here. And they're not quite slender and long enough, as seen in picture C, to be bacilli. So these would be called coccobacillus. A little bit of column A, a little bit of column B in this species. You should be able to identify these stubby individuals from their more slender bacilli if given an example in an image or under the microscope. Not all bacterium choose to be perfectly straight. Some bacterium have curves in them. If a bacterium has a single slight curve in it, or mild curve, it is referred to as a vibrio. If that bacterium has many curves in it and has this rigid corkscrew-like morphology, it is called a spirillum. Note the thickness of the wall in relationship to the length of the cell. Spirillum are relatively thick and spiral shaped. And this is important because it is not to be confused with a spirochete, which is thin and long. Because of this, they are springy. Carefully compare this to the stubby form of spiral bacteria, which are much stiffer in their design. Before we discuss the cell wall, we need to discuss one of the most important functions, the movement of chemicals in and out of the cell through the cell wall. Osmosis is the movement of water in and out of a semi-permeable membrane in order to balance out the chemical concentrations. By definition, a cell has a membrane that surrounds it. This membrane does not prevent water from moving in and out and is therefore semi-permeable. Water is too small to prevent its movement in and out of the cell membrane. If chemicals, like salts, are really high in concentration, in other words, the number of chemicals per volume of water is high, outside or inside of the cell compared to the other side, water will move either in or out to try to balance the chemical concentration. In biology, water has chemicals dissolved in it. We call such chemicals solutes, and we call water the solvent. Examples of solvents you may be familiar with in your household include the acetone and fingernail polish remover, mineral spirits, or ethanol. The most common example of a solute in nature are salts. Put salt in a cup of water, the water will act as a solvent and dissolve the salts, which become a solute. In example A, the ratio of solute to water molecules is relatively the same on the inside versus the outside of the cell. We call this isotonic. Tonic is the fluid surrounding the cells, so it's the external environment. In example B, the solute to water ratio is higher on the outside of the cell than inside the cell. So there's more salts or solutes in the water, the higher concentration of them outside of the cell. This is called hypertonic. Hyper means high. So there's a high amount of solutes outside the cell. Therefore, to counteract this imbalance, water rushes out of the cell. Because the solutes are trapped, cannot equilibrate by moving inside the cell. Water has to do the work here. And this dehydrates the inside of the cell as the water moves out until the concentration at some point of the solution inside the cell or the cytoplasm becomes isotonic, where the cells are destroyed through crenation. And crenation is just, as you can see, the cavitation of the cell as it's dehydrating out, like if you're taking the air out of a balloon or something. This dehydrates the inside of the cell to the point it becomes isotonic, and crenation can form. As water leaves the cell in hypertonic solutions, this can cause the cell wall to cave in, called crenation again, and too much crenation can cause the membrane to tear. This is called plasmolysis. Lysis means to tear or to break, And that cell membrane is called the plasmid membrane, so plasmolysis. This tearing can also happen when too much water enters the cell. This reverse process is called cytolysis or osmotic lysis. And in example C, we can see this take place. The solution around the cell is hypotonic, or low solutes outside the cell, and water rushes through the membrane inside the cell to try and create an isotonic solution, and it dilutes the solutes until isotonic solution is reached, or we have osmotic lysis or plasmolysis. And here we can see the movement of water. An isotonic solution, water's movement inside and out is equal to each other. There's no net change in the amount of water inside or outside of the cell. This is called homeostasis. And the cell's wall is unaffected. In a hypertonic solution, the water exits the cell to try to balance the concentration of salts. And finally, in a hypotonic solution, more water enters the cell to try and dilute the concentration of salts inside the cell. Checkpoint 1. How would we describe the morphology of the bacteria seen in the circled microscope image? A little note here, you'll see in the bottom right corner, it will say 20 micrometers. And that's what we call the scale bar on a microscopy image. It's referring to the non-zoomed in portion of this image. I created the zoomed in image circled in red. You'll notice that this chain of bacteria is made up of many smaller bacteria. You can see the individuals there. Okay, so remember, we're looking at sizes of about 1 to 2 microns, so keep that in mind, that this is a chain of bacterium, not one single bacterium. Checkpoint 2. How would we describe the morphology of the bacteria seen in the microscopy image? Now let's get into the different parts of the cell. We've already discussed the nucleoid a little bit, but there are also these regions called plasmids, which are extra chromosomal genetic elements. That is to say, they're not part of the chromosome. They don't exist in the nucleoid region. In fact, if we look here, we can see the little dotted plasmids are outside of this nucleoid region, and we'll discuss them a little bit more later. We can see in this image the nucleoid region. The dotted line outlines where it is. It is not contained within a membrane like eukaryotes, but it's kind of a condensed area of DNA found within prokaryotes. And these nucleoid regions are basically impossible to be seen with the light microscope. You can't see them with the transmission electron microscope. But if you're looking at a cell under the microscope, and you look, you can see the maybe a little area where the DNA is stored. You're probably actually looking at a nucleus in a eukaryote, not a prokaryote. So a little bit more about plasmids. Plasmids are much smaller than the chromosomes. They are very, very small, and they're always circular. Plasmids are like additions of DNA that help in special conditions, but not for basic biological functions. Examples of special conditions are for survival to antibiotic exposure or resistance to other sorts of stresses, such as temperature stresses. You wouldn't find genes on plasmids encoding for the production of the cell wall or binary fission, as these are genes that are essential and they will be found on the chromosome. Recall that Carl Woese used the ribosomal gene sequences to build the phylogenetic tree. So, what exactly are ribosomes? The ribosomes are the location where proteins are synthesized. Proteins are the tools or machines of the cell, and ribosomes are the machines that make the machines. Next, the cytoplasm is the goo or liquid portion inside the cell that everything floats within. Since each domain, eukarya, bacteria, and archaea, need to make proteins, the ribosomes are found in all domains of life, and they're mostly identified by their size. Instead of their size being measured in grams or meters, It is measured in a Svedberg unit. The size affects how fast they settle in a solution. And so ultracentrifuges, which is a lab machine that spins samples around really fast, accelerates the rate at which solids settle in a solution. The size of a ribosome affects how quickly it settles in an ultracentrifuge. This settling rate is called a Svedberg unit. Prokaryotic ribosomes that are found in the cytoplasm have a size of 70 Svedbergs or 70S. And this is going to be very important. It seems trivial right now, but these are going to be one of the little factoids that you will have to memorize when it comes, as it's going to come up more in other chapters as we dig deeper. So these ribosomes can be categorized by their size, and prokaryotes are going to share similar sizes, and they're going to be different than eukaryotic sizes. So all prokaryotes should have a 70S ribosomal unit. Inclusions are nutrient storage centers for the cell. Nutrients like glycogen, carbohydrates, starches, sulfur, phosphates, etc., are stored in inclusion bodies to be saved for a rainy day. Let's turn our attention to the cell wall. We'll start on the outside first. Some bacteria make a substance made out of different types of sugars. Not really the tasty varieties we all love and know. But notice here we have the glycocalyx. And glyco sounds like the sugar glucose. Please take some time again to review sugar, lipids, nucleic acids, and amino acids if you haven't already. You should already know how they vary chemically and the purpose of each of them. The glycocalyx literally means sugar coat, is a sticky substance that is secreted by bacterium and coats the outer cell. When it is well organized and tightly attached to the cell wall, it is more specifically called a capsule. The glycocalyx or capsule provides help or helps shelter the bacteria from the environment and from being attacked by others, such as immune cells. In this way, it helps protect the cells from the host's immune system and contributes to the cell's ability to be virulent. Virulence means the ability to cause disease. This is why the capsule stain is so important when identifying pathogenic bacteria. For example, only encapsulated Streptococcus pneumoniae cause pneumonia. An unorganized and loosely attached glycocalyx is endearingly called a slime layer. Slime layers allow cells to attach to each other, attach to surfaces, and to form what we call biofilms. Biofilms are very important in nature. 99% of bacterial cells exist in the form of biofilms. As seen in this image to the right, slime layers can also increase the virulence of organisms. For example, biofilms can form within a catheter, which prevents the organisms from being flushed away. It allows them to attach and grow on the inner surfaces of the catheter. This can also occur in other parts of the human body and therefore increase the virulence of an organism. Flagella are protrusions from the cell that can be used like propellers. As they spin, they propel the cell. Bacteria can sense when they are getting closer to a chemical source by how concentrated the chemical becomes as they move. This is called the chemical concentration gradient. And chemotaxy is the process of moving away or towards a source based on sensing chemicals. The same can be true for phototaxy. Uh, Cells that photosynthesize light may want to move toward the light source. As they move, they can tell which direction the light source is from by the change in intensity or concentration of the light. In these four images, we have different examples of flagella. In image A, we have something called peritrichus. Peri means around, so we can find flagellum all the way around the cell. In image B, we have monotrichous. It's called monotrichous not because it's on the end here, but because there is a single flagellum. It is also called a polar flagellum because it is at one of the polar ends of the bacterium. For a bacillus, polar would be at one of the ends. That's pretty easy to tell here that it's not in the midpoint. It's important to follow the definitions of the book and lecture because you might find other definitions by doing a web search. The next two are lophotrichous, which is a tuft of flagellum in one spot, and amphitrichous, which means that there are flagellum located at two different locations. Here they are amphitrichous and polar bacterium. Fimbriae are short appendages that adhere to other cells and surfaces to assist in forming biofilms. And here we have pili, which are similar in structure to Fimbriae, but used for twitching motility. So basically, these pili, they don't work as a propeller; they twitch in order to help with movement. And they are very importantly a special tool for the transfer of plasmids between different cells. And this transfer of genetic information is called conjugation in bacteria. This is what it looks like. In this image, we can see a pili connecting two bacterial cells. And this is how a bacterium can transfer plasmid DNA from one cell to another. So there's a tube, a hollow tube in this pili where the genetic material is moved. And so plasmid can be transferred from one cell to another, and this is called horizontal gene transfer. And it's called horizontal gene transfer because we're not creating a offspring here. It's just through two adult bacterial cells. And that's what they mean by horizontal. If you look at a familial tree, right, everything that's horizontal in a family tree are going to be from the same generation. So that's the figure of speech that's used here. An example of vertical gene transfer would be how humans transfer genetic information, which is from offspring to from parent to offspring. These two are just exchanging information without creating any new cells. This transmission of plasmids is very important to bacteria. As previously discussed, plasmids have non-essential genes that allow them to survive certain stressors. One of these can be antibiotic resistance. So, if one bacterium has a resistance to an antibiotic, that gene can quickly be spread throughout a population through this method of conjugation. Now, there's no need to memorize this information in this slide, but scientists are now using plasmids to fight fire with fire, and they can use artificial plasmids to be introduced into a bacterial population. And these plasmids can outcompete the native plasmids. And importantly, these artificial plasmids lack those antibiotic resistance genes. And therefore, it can weaken the bacterial population and make it more susceptible to our treatments. Checkpoint 3. How would we characterize the flagella of the bacteria seen in this microscopy image? Bacteria have many layers. Working our way from the capsule in, and remember, not all bacteria have a capsule, is the cell wall. The cell wall is made up of different components. It rests on top of the plasma membrane, which is the innermost membrane. Here is an electron microscope image of what the cell wall looks like, and so it's labeled as CW. It rests on top of the plasma membrane and helps shield it from the external environment. Its rigidity resists the tendency of the inner membrane to want to burst apart in hypotonic solutions. The cell wall is also what anchors the flagella and other important proteins for cellular function. The cell wall contains peptidoglycan, We've covered this a little bit before, but peptidoglycan is a complex material and is composed of rows of disaccharides linked by polypeptides. The disaccharides are made of two sugars, and polypeptides are proteins made of several amino acids. Again, cannot stress this enough. You should review these different biomolecules of life. We will talk a lot about gram-positives and gram-negative cells. They each have cell walls with unique compounds and unique spatial arrangements that you will need to memorize. Gram-positive cell walls have much thicker peptidoglycan layers on the outside of the cell wall, pictured on the top in the picture. They also have two acidic compounds called teichoic acids and lipoteichoic acids that penetrate them. Lipoteichoic acids go all the way through and into the cell membrane, which is why they're called lipoteichoic acids. The peptidoglycan makes the gram-positive cell walls thick and rigid. Remember that the crystal violet iodine complex in a gram stain is really hard to remove from the cells, and that's why they remain purple after staining or after destaining. And this is due to the thick peptidoglycan layer that gram-positive bacterium possess. We also have what we call the plasma membrane underneath the cell wall. The plasma membrane is a lipid membrane. Teichoic and lipoteichoic acids improve the stability of the cell wall. Teichoic acids are only cell wall deep, whereas lipoteichoic acids again penetrate all the way down and into the plasma membrane, also known as the inner membrane. Both gram-positive and gram-negative bacteria contain plasma membranes, and they also have porins, which are small membrane-bound proteins that act as channels for moving things in and out of the cell. The electron microscope image shows how thick the peptidoglycan layer can be, and then this thin cell membrane inside of it. Gram-negative cell walls have a thinner peptidoglycan layer, fewer layers built upon each other. The peptidoglycan is sandwiched by two membranes, the outer membrane and the inner membrane. That inner membrane is again called the plasma membrane, and gram-positive bacteria have it as well. The space between the outer and plasma membrane is called the periplasmic space, or the periplasm. It is the region where the peptidoglycan is found in gram-negative bacteria. Because gram-positives lack an outer membrane, they do not have a periplasmic space. Note, there are no teichoic or lipoteichoic acids in gram-negative bacterium. However, there is another substance unique to gram-negatives, that is the lipopolysaccharides, abbreviated as LPS, that is embedded in the outer membrane. You must memorize the order of the cell wall of gram-negative bacteria. So starting from the inside, working our way out, we have the plasma membrane, the periplasmic space, the thin peptidoglycan layer, and then the outer membrane, which has LPS embedded in it. And here is the electron microscope image of a gram-negative membrane. For reference, a bacterium is about 1 micrometer wide, and that's about 1,000 nanometers in a micrometer. The width of the inner membrane, or the outer membrane, both, is about 10 nanometers, or 1/100th the width of a bacteria. Note how the outer membrane shields the peptidoglycan in the gram-negative bacterium. Also, the peptidoglycan doesn't interface with the outside world in the gram-negative bacterium like the gram-positive cells do. This means that peptidoglycan, while important, isn't as critical with the gram-negative bacteria. Some antibiotics are developed to attack the peptidoglycan. And based on what we discussed, why are gram-negative bacterium less harmed by these antibiotics? The reason is their role is less essential in maintaining the integrity of the inner membrane, and it's partially shielded by the outer membrane. As a warning, you will be required to be able to draw these images out. Now, you don't have to make them look pretty like we have here, but I want you to be able to maybe draw some lines, very distinct lines, and label them like they're labeled in this image here. So a line for the inner membrane, labeling it as the cell membrane or inner membrane, a line for the peptidoglycan, label it as peptidoglycan, so forth and whatnot. I want them distinct, so don't draw them so close together that you try and fool me. Sometimes that happens. We don't want to create a sense of ambiguity here. Um, you want to have clear lines and clear labels so that I can tell that you know what you're talking about. Checkpoint 4. Gram-negative bacteria do not have blank. And checkpoint 5. Penicillins are an effective antibiotic against gram-positive bacteria. They disrupt the structure of peptidoglycan. Penicillins are much less effective against gram-negative bacteria. Why? Acid-fast cells have yet another chemical called mycolic acids outside of their cell wall. These waxy substances help protect and shield the cell, similar to how the glycocalyx works or the capsule works. Examples of organisms that contain this are Mycobacterium that can cause tuberculosis and leprosy. We've already kind of discussed the plasma membrane. The plasma membrane is the membrane that houses the cytoplasm and all of the cell's inner contents. It's a selectively permeable membrane or barrier that encloses the internal structures of the cell. So essentially, the plasma membrane acts as the gatekeeper that decides what sort of nutrients or products go into the cell and what sort of toxins or other things are excreted outside of the cell. Some species of bacteria can turn from normal and active cells, which are called vegetative cells, into endospores. These are relatively inactive cells that have turned into a sort of bomb shelter that is resistant to environmental extremes like high heat and radiation. They do so to survive harsh conditions in order to emerge as a vegetative cell at a later time and continue on their merry way. This table shows the different traits of each type of cell. Vegetative cells tend to be sensitive to extreme temperatures and radiation, whereas endospores are resistant to extreme temperatures and radiation. Vegetative cells are going to stain as gram-positive. However, endospores don't absorb gram stain, and so as a result, you'll need a special endospore staining technique. Vegetative cells have normal water content, and the enzymes that carry out chemical reactions inside the cell have a normal activity. Endospores are much more dehydrated, and there's a very low, in the book it says no metabolic activity, but it's so little that they just refer to metabolic activity, probably to help you differentiate between the two. So there's metabolic activity with vegetative cells and no metabolic activity with endospores. Vegetative cells are capable of active growth and metabolism. And as you can imagine, endospores are dormant, so they don't experience growth or metabolic activity. And these endospores also resist chemical penetration very well, hence them not taking up the gram stain very easily. The process of becoming an endospore is called sporulation. The process of returning back to a vegetative cell is called germination. 6 basic steps are involved in the transition to becoming an endospore. First, the DNA is fortified with membranes. The precursor to the spore is called a forespore that is formed. This forespore builds more membranes around it. Then a shell called a cortex is formed around them, and a protein coat goes around all of that. Then finally, it is released from the cell to become its own individual spore. And we won't cover germination in detail in this class. Spores can hang around for a very, very, very long time. And in this research, they analyze what is called the ultrastructure of spores. And that's just what ultrastructure just means the subcellular level. So, what's going on inside of the cell. And what they found were intact bacterial spores in the skin of an Egyptian mummy. And they showed that the bacterial spore had all of the characteristic appearances of a spore. So they still had the core, a cortex, a coat, and an exosporium, which is just the outer layer of the endospore coat. And that will wrap up Chapter 3, Part 1. Stay tuned for Chapter 3, Part 2. Thank you all for watching, and please watch the second half of Chapter 3.